Yeah, well, hey, thank you, Will. Thanks for running this meeting. I'm Michael Lynch. I'm the CEO of Chonex. I've been working very closely with Lynn Rogers for over 5 years. Lynn has brought a depth of knowledge and experience to understanding the biology in our product and how it interacts with the biology in the soil. I know she was trained by Elaine Ingham and really understands the soil microbiology and the soil web. And she's done a great job educating me, educating my board, educating my investors, and helping us really understand the functional groups, how those microbes grow, the concentration rates of those microbes, and then how to best apply them. And helped us develop our product over the last 5 years. And it's an honor to have you here, Lynn. And I'll turn it over to you. And I really appreciate all your help and are grateful to have you here with us today.
Once again, thank you, folks. Thank you, Michael. I know for most of you, the growing season has already started, and you're all very, very busy. I do really appreciate your time attending the webinar. Let me tell you a little bit about microbial matrix systems. Once again, Lynn Rogers, and microbial matrix systems were located in Albany, Oregon. And for the past 23 years, I've collaborated with farmers trying to create microbiome programs, that is, programs utilizing biostimulants, biologicals that farmers can integrate or combine with their existing fertilizer program, their existing herbicide program, their existing pesticide program in order to enhance what I call fertilizer use efficiency, nutrient uptake, and also improving soil structure, resulting in a boosting of crop yields. During those 23 years, it became apparent to me that assisting the manufacturers like Chonex Ag in the testing of their biostimulants or biologicals to ensure product quality is just as important. So I've had a great experience of working both with farmers and with manufacturers, resulting in, I would say, a greater appreciation of why biologicals or biostimulants are important in production agriculture today. So I'd like to talk to you today about how we looked at the different mechanisms of actions of how StrongSoil helps to enhance nutrient acquisition and efficiency, resulting in better soil health and crop production. One way we do that, we offer a laboratory service called the functional group assessment. Basically, functional group assessment identifies the bacterial groups that improve nutrient absorption, maximize fertilizer effectiveness, and of course, improve crop production. So it's a starting step to trying to understand the mechanism of action of how a biostimulant or biological works. We basically look at 7 groups in the functional group assessment method, and I'll just go over those 7 groups real briefly. Throughout the presentation, we'll dive deeper into the science behind these 7 groups. The first group that we really examine is the endospore-forming bacteria. They are a group that is comprised primarily of the genera Bacillus or Panabacillus. Endospore-forming bacteria are very, very important because they help to improve soil structure, nutrient acquisition, and mitigating drought and heat stress. We also look for free-living nitrogen-fixing bacteria in strong soil, and free-living nitrogen-fixing bacteria fix atmospheric nitrogen, i.e. N2, and convert it into ammonium or ammonia. We also look at phosphorus-solubilizing bacteria because this is a very important group. In different parts of the country, whether you're in West Texas or even different parts of California, there could be a lot of phosphorus in that soil, but that phosphorus is locked up tight. It's combined to various soil elements like calcium, for example. So strong soil has a significant amount of phosphorus-solubilizing bacteria that can help solubilize that phosphorus, not only freeing up that phosphorus, but also freeing up uh, micronutrients in the soil as well. Pseudomonads is one of my favorite group because it uh, participates in root, canopy, and seed development. There's still other pseudomonads that are very great at mitigating drought and salt stress. We also look at heterotrophic bacteria because they also play a role in soil structure, nutrient acquisition, and plant growth. I want you to also realize that when we're plating out, when we're assessing during the methodology, a lot of heterotrophic bacteria will also end up being on the same agar plate as endospore-forming bacteria. Cellulose degraders, again, we look at because they do help build soil organic matter. And finally, the seventh group is chitin utilizing bacteria. This is a great group to have in a biological because unfortunately, many plant fungal pathogens, their bodies are made up of
Claro,
chitin.
todo y sigue saliendo agua.
So having chitin utilizing bacteria can help to manage a farmer's disease and pesticide program. So let's start diving deeper into each of these individual groups. Endospore forming bacteria, when deprived of food, water, and oxygen, these bacteria become dormant and turn into what we call an endospore. You can see this chain of cells already forming an endospore as opposed to this chain of cells where it's still in its more vegetative and active growing form. Why are endospore forming bacteria important? Well, let's talk a little bit about what they are. Endospore forming bacteria, as I just mentioned, have the ability to create a structure called an endospore. That endospore structure is a protective mechanism during extreme periods of heat, drought, or even nutrient scarcity, lack of oxygen in the soil, and even high salt conditions. But also, an endospore forming structure is a protective mechanism during the production process of many biostimulants. It helps protect the bacteria from drying out, from drying and lack of moisture, and maybe low levels of oxygen during the production process. Different soil types may have high or low levels of nutrients. Obviously, if you have a very low level of nutrients, then you're going to push these endospore forming bacteria into a dormant, non-active form. Also, different parts of the country, we have very compacted soils, soils that may have very low levels of water infiltration and oxygen. But endospore forming bacteria are a very important group because they do play a significant role in improving soil structure. They also are responsible for nutrient acquisition and mitigating heat and drought stress. So how do manufacturers like Chonex Ag deliver? Obviously, in many parts of the country, the endospore forming bacteria are either non-active or they're simply dying off because of certain soil conditions like compaction, for example. As Michael had mentioned, over the 5 years, I've helped Chonex Ag look at the different matrices or formulations of strong soil. We looked at their insect frass. Again, just over the years, talking together, they have formulated a water dispersible granule and more recently a wettable powder form of strong soil. But what I wanted to talk to you about today is, folks, they have been able to retain endospore forming bacteria through their different processes and matrices of strong soil. So now there's a way for manufacturers like Chonex Ag to deliver an active endospore forming bacteria. This is just an example. We do a lot of trials and tests of strong soil. So when you add strong soil to a fertilizer tank, you have now oxygen, you have agitation. As you can see here, you have fertilizer, so nutrients for the bacteria, the endospore-forming bacteria, to become active. In fact, the endospore-forming bacteria found in strong soil does increase over time while it is in the fertilizer tank. You see here a lot of foaming, and that foaming indicates endopolysaccharide production. And as you know, exopolysaccharide production plays a role in nutrient uptake, moisture retention, soil structure remediation, and even protection of the plants from drought and salinity. So now Chonex Ag, because of the production process, has been able to activate this population of endospore-forming bacteria. When applied to ag soils, these bacteria are able to hit the ground running and participate, as I mentioned, in these different soil processes. Just wanted to take a moment and show you some of the auger plates that we look at when we assess strong soil. The first row is the heterotrophic bacteria, and the bottom row represents the endospore-forming bacteria. You can see whether we go from the different matrices or formulations of strong soil, we're looking at the same dilution. So it's, in other words, the retention of heterotrophic bacteria is actually retained just no matter what the matrix or form of strong soil is in. The wettable powder, as you can see here, I probably should have been counting the 10 to the minus 5 dilution because it would be easier. There would be less colonies here. It would be a lot easier to count. But just for demonstration purposes, you can see because the wettable powder has a smaller surface area, you actually increase the colony forming units of the bacteria. Also in the endospore forming bacteria plates here, we got the frass, the WDG, and the wettable powder. And as you can see, the population or colony forming units is also retained throughout the different formulations of strong soil. Again, wettable powder, smaller surface area, higher colony forming units. So Chonex Ag has been able to, through the production process, retain a good bit of their endospore forming bacteria, activate it so it can, upon application, hit the ground running, getting ready to participate in a variety of soil and plant processes to promote plant growth. But also, there is a mechanism called plant microbe interactions where those endospore forming bacteria, once applied to the soil, can continue to stay active and possibly increase their population in soil. Why? Simply put, the plant, whether you're looking at corn, soybean, cotton, blueberries, that plant provides a food source, either sugar or protein, through its root system to the bacteria. So the bacteria get a food source from that plant, from the plant roots. In return for that food source, the microbes provide NPK, iron, zinc, etc. to the plant. So it's a mutualistic relationship between the plant and plant growth promoting rhizobacteria or PGRs. So endospore forming bacteria, as I mentioned, plays a very important role in improving soil structure. Improving soil structure results in better nutrient acquisition. For demonstration purposes in the lab, we took a petri dish, we added sodium chloride, salts. We also added some sandy soil particles and some clay particles. We dropped in a biological, and in about 3 to 5 days, we start seeing all of these particles attached together. And the reason being is bacteria produce exopolysaccharides that simply act like glue and glue these particles together. So if you can envision this as your soil, when this happens, when the exopolysaccharides are produced and these particles are glued together, you have these pockets. Now you have better water penetration, pockets for better water infiltration, as well as pockets for better root penetration. When that happens, those plant roots now can meander through these different pockets, getting access to water, getting access to fertilizer and other plant needed nutrients. Also, look at the salt particles being glued together. That's a good thing in many soils throughout the country where high salt is a huge issue. The salt particles are now glued together, preventing any uptake of sodium and chloride that can affect or hinder plant growth. Also, in this particular biological, there is fungi. Fungi tend to grow in what we call hyphal strands. And you can see that fungi here. You can see that hyphal strand starting to grow and reach out to soil particles. In about 2 weeks or so, that hyphal strand will start connecting all of these particles together. But still, we have these pockets, these spaces here for better water infiltration, again, better root penetration. So plant roots can now have access to plant needed nutrients to water. Also, you're holding more moisture in that soil. By having these pockets, now you have a more aerobic, tilthy soil. And why is that important? When we even look at fertilizer like urea, urea needs to go through an oxidative process. In fact, plants do not absorb that urea. That urea is converted into nitrate nitrogen through an oxidative process. So having very good improved soil structure and an aerobic, tilthy soil structure results in better acquisition of water, plant needed nutrients or fertilizer, and as well as supporting the endospore-forming bacteria. Free-living nitrogen-fixing bacteria we grow on what we call minimal nitrogen agar plates. But one thing that is interesting about StrongSoil and is unique, I think, to other biologicals and biostimulants on the market is StrongSoil is involved in the nitrogen cycle in 3 areas of the nitrogen cycle. They do have free-living nitrogen-fixing bacteria, i.e. azospirillum. There are some endospore-forming bacteria like Bacillus megaterium that also can fix atmospheric nitrogen. Many of the endospore-forming bacteria in StrongSoil can take nitrogen from organic sources and convert it into ammonia or ammonium. Still, other endospore-forming bacteria, which Chonex has, called Panabacillus, can, through a nitrification process, convert that ammonia or ammonium into nitrate and eventually, or nitrite rather, and eventually into nitrate, which, as we said, is the form that the plant can assimilate. So very interesting biological, and most biologicals in the market may only have endospore-forming bacteria that can participate in the nitrification cycle, or they just have an azospirillum or something like that that fixes atmospheric nitrogen. Not to say that those products are any less better or any more better. It's just that this is characteristic of StrongSoil throughout our 5 years of assessment and testing. StrongSoil also has a high amount of phosphorus-solubilizing bacteria. In the laboratory, we use calcium triphosphate, and you can see here the 3 plates made up of calcium triphosphate and some other nutrients. When we inoculate these plates with StrongSoil, you'll notice here is what we call a zone of clearing, and that's what we look for because that indicates that in StrongSoil there are phosphorus-solubilizing bacteria that have an enzyme that can easily cleave the bond between calcium and phosphorus, now making phosphorus much more readily available. And again, this is really, really important because in different parts of the country, phosphorus is bound or tied up to calcium. Phosphorus-solubilizing bacteria is also very important because not only can it be bound to calcium, it can be bound to a lot of different elements in soil and therefore unavailable to plant roots. And depending on where you are in the country, whether you're in Texas or California, calcium can tie up phosphorus. You go to the Midwest, Iowa and Minnesota, Kansas, magnesium and manganese can tie up phosphorus. Here in the Pacific Northwest, we have very acidic soils, so iron and even sometimes zinc can tie up a lot of our phosphorus. So that phosphorus is not readily available to plant roots. In fact, many farmers can add more phosphorus in their fertilizer, even though they have a significant amount of phosphorus in their soil, but it's not soluble or made available. But having phosphorus solubilizing bacteria that have the enzyme, and bear in mind, there are many mechanisms that phosphorus solubilizing bacteria can do to or can use to help solubilize phosphorus. I'm just talking about one simple mechanism here, and that is the bacteria that have an enzyme called phosphatase. They simply cleave the bond between that substrate, between calcium, between magnesium, making phosphorus now more soluble and easily acquired by plant roots. Also, in different parts of the country, you have different soil pHs. Soil pH can affect the availability of phosphorus to plant roots. So looking at a pH graph here, starting out on the acidic side, which is up here in my neck of the woods, we have very acidic soils, and phosphorus tends to be fixed or tied up or locked up tight to iron. As we move along the pH curve, maybe to areas like Kansas, for example, or Iowa or Minnesota, their pHs are leaning more towards a high acid range or more towards a pH range. And we find that phosphorus can be bound to and not available to things like magnesium, aluminum, manganese. Then if we meander down to areas of California, to West Texas, for example, they tend to have high alkaline, high calcareous soils. So phosphorus is tied up with calcium and not readily available. Even if a farmer or grower may add more phosphorus to their fertilizer and then to the soil, that phosphorus ends up being tied up by calcium. Again, not readily accessible by plant roots. So having phosphorus solubilizing bacteria in a biological is very, very important, again, to help solubilize and make phosphorus much more available to plant roots. Now, we talked about nitrogen, we talked about how nutrients are easily solubilized, phosphorus is easily solubilized and made available to plant roots. But the key to nutrient acquisition success is not just making sure that those nutrients are available. It is making sure that those nutrients are acquired at a specific plant growth stage, a nutrient demand stage. So as for demonstration purposes, let's just look at corn. Corn has various plant growth stages. When we look at V4 to V7, that demand for NPK starts going up as we approach V7. Okay, so very important to deliver nitrogen. The bacteria need to solubilize and make nitrogen, phosphorus, and potassium available before V7. As we move from V7 to V12, for example, notice that sharp demand, that sharp increase for NPK, more so for potassium, but definitely nitrogen and then phosphorus. Of course, that demand continues through tasseling and into kernel fill. But what's important here, again, just for demonstration purposes, Look at the orange curve. That's going to represent the high nitrogen demand as we go through V7, V12, and tasseling of corn. But because of maybe certain environmental conditions, you can't control the weather. You may have extreme heat temperatures increasing. And so when a farmer or grower drops in their fertilizer, their urea, you can inadvertently have early nitrification. So that nitrate nitrogen actually peaks and becomes available before the peak demand for nitrogen. You can be in Kansas, for example, and have excessive early rains resulting in denitrification or the conversion of ammonia into nitrite nitrogen, for example. So you're not actually making nitrogen available at peak nutrient demand times. But having biologicals like StrongSoil, what they do, whether it's the free-living nitrogen-fixing bacteria, the endospore-forming bacteria that participate in ammonification and nitrification, they go through a process I call banking. They simply hold that nitrogen in their bodies, and as we move through the growing season, they then release that nitrogen, thereby synchronizing these two curves. So nitrogen now is much more efficiently available to plant roots. You can look at any of these nutrient demand curves, not just corn. There's the same nutrient demand curves for cotton, for example. Pseudomonads is another group that we assess for. They're very easy. We use King's Bee medium, and many pseudomonads will produce a fluorescent pigment. So when we put a black light over the plate, you'll see this fluorescent molecules here that the pseudomonad is producing. There are still other very, very beneficial pseudomonads that don't produce the fluorescing pigments. In this case, this is Putida. Of course, we would probably submit it to a laboratory that would test for DNA to confirm, but just over 23 years of doing this, I would know that this is Pseudomonas putida. It has a very significant cellular morphology. You have this raised, wrinkled, almost eggshell white color. The chances are Pseudomonas putida is what those colonies are. So Pseudomonads are a very important group to have in any biological. One of the important reasons is because many Pseudomonads produce IAA, or what we call indoacetic acid, which is a precursor to plant growth hormones called auxin. Auxin is a hormone needed for better root development. Still, other Pseudomonads can produce plant growth hormones for better canopy and seed development. Many of the Pseudomonads, like Pseudomonas putida, for example, can help to mitigate heat and drought stress. They can also, like Pseudomonas fluorescens, trigger an ISR response in plants. ISR is just induced systemic response. I like to think of it as during the cold and flu season, we tend to eat all of our probiotics, eat our yogurt, drink a lot of orange juice, just to boost our immune system during that cold and flu season. Also, Pseudomonads can help to improve nutrient acquisition, and they do this by producing compounds called siderophores. Siderophores help to chelate iron. These are some photos from Minnesota. You have very clayey soils. In parts of Minnesota, you may have a lot of early rain during the season. So it affects soybean production. You end up having a lot of what we call iron deficiency chlorosis in soybeans. And because of the clay soil and excessive rain, it results in a lot of wet, compacted soils and lack of oxygen, a buildup of CO2. So much so that the bicarbonates found now in this soil condition kind of prevent or dilute or neutralize the acids that are normally produced by soybeans to chelate or solubilize iron. In Minnesota, iron is in abundance in their soil, but it's also in a form called ferric iron or F3 plus. And that is the form that plants do not take up. The plants need the reduced form of iron, which is the ferrous state or Fe2 plus. And that is the form, just like we talked about nitrate nitrogen being the form that the plant can assimilate or absorb. The ferrous state of iron is what the plant can easily absorb or acquire. And also that ferrous state of iron easily moves or what we call translocate through the plants. So the bacteria, the Pseudomonads found in strong soil can produce these siderophores, chelate it, bind to it, and reduce it so that it can move more easily throughout plant roots and the rest of the plant. Heterotrophic bacteria, we use tryptic soy agar. Heterotrophic bacteria obtain their energy or food source from organic compounds. So hence, that's why we use an agar plate called tryptic soy agar. But what I wanted to point out to you folks is that many of the heterotrophic bacteria that we found in strong soil are also beneficial endospore-forming bacteria. This is an example of Bacillus subtilis. To the right, you have Bacillus pumilus. Again, we look at both groups just to verify that you have a certain population of endospore-forming bacteria where they can easily obtain their energy or food source from organic compounds. Cellulose degrading bacteria, again, help with building up a soil organic matter. We use cellulose plant material. Again, just like the nitrogen process or the phosphorus-solubilizing bacteria, these bacteria contain yet another enzyme called cellulase. And if the bacteria in strong soil are cellulose degrading bacteria, you'll see what we call a zone of clearing. And we would know that that particular colony is a cellulose degrading bacteria. We finally come to the chitin utilizing bacteria. And again, we're looking at yet another enzyme that the bacteria in strong soil need to have, which is chitinase. If the bacteria have chitin, and we take an agar plate that is simply made up of crab shells and shrimp shells. Why? Because their bodies are made up of chitin. But if the bacteria I shouldn't say if, there are chitin utilizing bacteria in strong soil that have that enzyme and they will start metabolizing and eating that chitin, again, resulting in yet another zone of clearing. So very easy to determine that strong soil does contain chitin utilizing bacteria. And again, a very important group, a very important mechanism of action that a biostimulant or biological should have, because having these population can help a farmer with their fungicide or pesticide program, help to improve the overall efficiency. So we talked about all of these 7 groups, all of these 7 mechanisms of action, if you will, that strong soil can have to help improve soil structure, help to acquire nutrients, help to promote plant growth. But how do we know that strong soil is going to work in Oregon? How do we know it's going to work in cotton in West Texas? How do we know that it's going to work in soybeans in Minnesota or in Alabama? I wanted to talk to you a little bit about yet another plant microbe interaction. And it's actually a plant microbe communication system, and that's quorum sensing. Quorum sensing is simply a system of communication between bacteria, between the environment, and plants based on the population density of bacteria. Bacteria produce what we call autoinducers. On the bacteria, there are receptor sites that these autoinducers can combine to. when the bacteria then receive those signals, receive those autoinducers, it'll coordinate their behavior and activities with other bacteria. And so, for example, a signal is received by that bacteria to tell yet other bacteria to start producing exopolysaccharides for biofilm formation. Maybe it can trigger a signal for the bacteria to start congregating or aggregating, to start producing chelating compounds in order to chelate iron, like we just saw. Perhaps it's a signal for rhizobia to start regulating nodulation. Perhaps it's a signal for triggering nutrient uptake. So plants also respond to quorum sensing, which triggers possibly defense mechanisms like ISR being triggered by uh, uh, Pseudomonas fluorescens. Plants also respond to these signals, which can also trigger mechanisms within the plant to overcome drought stress. So in order to just kind of uh, give an example of quorum sensing, I'm going to use blueberries, for example. This is a farm in Oregon, and it was a 5-year-old blueberry farm, and a 5-year-old blueberry farm should be producing um, about 7.5 tons to the acre. But on this farm, there were areas where we see very nice bushes, obviously producing that 5 ton to the area. But there were spots throughout the field where the soil type was different. It was very compacted, very high clay content, resulting in wet anaerobic conditions, which triggered Phytophthora root rot. So even though the farm is 5 years old and the expected yield is 7 ton to the acre, it was not producing that. It was more like 3 ton to the acre. So having implemented a microbiome program, what we can do then is to help mitigate that soil structure, and also help to be that biological to have an antagonistic effects to Phytophthora and also elicit plant growth. And the reason why I wanted to use a perennial crop is even with the following year, you can see no matter where you are in this field, you're not looking at 5-year-old good producing shrubs. You're looking at the same formation, the same type of growth. So the point is that what you can expect from a biological or biostimulant is simply to raise the areas of a poor producing field up to the same area of the good producing field, i.e., giving the grower a yield kicker. Not that you can't contribute any more to crop production in the good producing fields or areas. You probably are maybe triggering a better quality yield that is, contributing, in this case, blueberries, maybe firmness, fruit firmness. But the important thing is now you've remediated the areas where there is poor production. Other laboratories that we use to not only assess, and bear in mind, you can use the functional group analysis to analyze what the mechanism of action in a biological could be. You can also look at using those tests to see if upon the addition of a biological, you are improving those 7 groups in your soil. We also have another slew of laboratory services called direct microscopy. And what direct microscopy simply involves is identifying the active microbiome of soil, compost teas, and biologicals and biostimulants. We look at how active the bacteria are, that is metabolically active. If that bacteria is active, it'll have yet another enzyme called dehydrogenase. And when we add in a fluorescein diacetate, and the bacteria have an enzyme that can cleave that bond between fluorescein and the acetate because it wants the acetate carbon portion for a food source, then the bacteria under the microscope will appear in this apple green color. We also assess for the amount of fungi in your soil. You can see here the little dots are bacteria, the bigger guys swimming around, that's protozoa. We assess for protozoa because protozoa are good indicators of soil moisture retention, water penetration, and they're also a unique set of organisms because if there is excess nitrogen, it'll make that more easily available or acquired by plant roots. We also look for beneficial nematodes as well as plant parasitic nematodes and mycorrhizal fungi. The reason why we assess for mycorrhizal fungi, it's not only just improving root development, but also finding a way to enhance the soil structure. In this case, you see here on the upper left hand, arbusculum mycorrhizae, they will shoot out these hyphal strands. Now you're getting a bigger root surface area, if you will. So now you've got a wider range or wider area now for that root to obtain water, to obtain nitrogen, phosphorus, and calcium. So microbial biomass is also key to nutrient acquisition, and we do assess soils for that because when you look at soils, generally up to 5% of the total organic carbon and 2 to 6% of organic nitrogen in soil is in that microbial biomass. It's in those endospore-forming bacteria, it's in the fungi, and it's in the protozoa. Before I leave you today, I want you to keep in mind that the full yield potential is established when the seed is sowed, and it will diminish during that growing season. Why? High heat conditions, drought conditions, salinity, plant pathogens. It can reduce that yield potential maybe down to 80%, 40% if you have severe plant pathogens or diseases. But today in modern agriculture, by utilizing biologicals and biostimulants, we can preserve a significant portion of that potential. So I want to thank you today for attending this webinar. Have a great growing season, everyone. Up on the slide, you can see contact information, email, and cell. So
Lynn, that's
I think we have time to open it up for questions.
Yeah, we have about 10 minutes for questions. If you have questions, please either put them in the chat or come off of mute or raise your hand, and we'll take them in order. I do want to sort of tee us up with kind of a few questions that I've been sort of thinking about as you went through this presentation, Lynn. One that just came up. You talked about sort of endospore forming bacteria and their ability to sort of go dormant, so to speak, and survive during conditions of where there might be lack of water or oxygen or food. Do you have any sense of how long endospores can last? And folks can keep track of those.
Yeah, they can survive for a relatively long period of time. I don't really have a good idea of how long, but they can survive for a relatively long period of time. But the question really should be asked in ag soils when there's constant tillage, when there's just over and over again of the same monoculturing system, there's nothing wrong with that. A lot of those spore formers may not come back. They may not even come back in a biological product. Their numbers may go down. So you need to have the ability to activate what you have, stimulate that activation, and then inoculate it in the soil. And I think that's where you kind of need to focus is They may be in different parts of the country at very, very low levels.
Hmm.
So you need products like Strong Soil to just inoculate and boost up that population. That's not to say, because the next question a farmer would ask, well, I've inoculated Strong Soil once, and sure, the endospore forming bacteria can survive for certain periods of time. We don't necessarily know, and you're not going to change. You can't. It's unrealistic to change any of the farming practices, and you can't control weather. So you're going to constantly hammer the activity of those endospore forming bacteria. So no, it's not applying Strong Soil once and you're all good. You do need to do repeated inoculation.
That's great, Lynn, and I'm making you, I'm pinning you for everyone. So just FYI, so I think this is really great. Larkin Martin has posted in the chat, and then I believe Guillermo, you have a question after this, but Larkin has mentioned one claim made in the sales pitch of enhanced quote unquote nitrogen fixing microbes is that natural nitrogen fixing microbes uh, do not make the effort to produce nitrogen when nitrogen is present. So they do not produce N in the presence of farmer applied N. Is there much truth to this claim? There's sort of a lot of kind of like claims that are going on out there that are hard to understand. So can you clarify that about, how nitrogen producing bacteria function in the presence of, nitrogen fertilizer?
Right. Right. I mean, in the scientific literature, we did see free living nitrogen fixing bacteria not perform well when there are extremely high levels of nitrogen, even phosphorus available. But there are many different genuses and strains of free living nitrogen fixing bacteria. And we've had the pleasure of working with a variety of different companies where that's for that specific free living nitrogen fixing bacteria, it's not necessarily a problem. And even with azospirillum, I do find that people do have success. They don't necessarily tank mix, but they can have another method or timing of action. But there's still other free living nitrogen fixing bacteria like the gluconoacidobacter doesn't seem to be a problem tank mixing it in the fertilizer tank. Even tank mixing it with herbicides it's not detrimental to that specific free living nitrogen fixing bacteria.
Great. So looks like there's a lot of detail in that question. So I think it depends on the strain is what I'm hearing. That's
Yeah.
great. Zuhair asked, and again, if I don't get these questions right, please come off of mute. But we wanted to know, Zuhair asked if you have assessed or measured the phosphorus desportation process rate or percent desportion process. I'm not sure if I'm getting that word right. So check the chat following the application of your biostimulator. So does that make sense to you, Lynn? Do you know how to answer this question, or Zuhair?
Yeah, I don't understand what they're asking. The desporulation.
Yeah, Zuhair, do you want to come off of mute and ask that question? Um, if not, we'll try to get that followed up. And Guillermo, I think you wanted to ask a question. So if you want to come off of mute and ask a question, I think that'll be great. And we have time for maybe one more after this. So Guillermo, go ahead.
Yes, I wanted to ask your opinion on the efficacy of endospores after they've spored, and you kind of already answered, but that's one of the ones I have written down. Compared to a fresh batch out of a wonderful bioreactor system, would there be a great difference in the next generations of endospores compared to a fresh new batch of, what you got because you made it?
Oh, so I think I understand your question. So, for example, let's say I put the strong soil in a fertilizer tank and I didn't finish applying. Maybe I don't get through it. I have 20,000 acres, for example. or more, and we're not done with the application. Do those endospore forming bacteria that are now older, for example, in the fertilizer tank, do they have the same efficacy as a fresh batch? And because of the many data, field data collection, I don't see a difference. You still see a positive plant response between quote unquote a fresh batch of endospore forming bacteria or a batch that's 3 or 5 days older. So
Yeah. I actually
it
meant
has
this
to be because farming
the ones in
operations, you may not be able to get through a
the whole
farm.
thing.
Yeah.
Yeah. I meant the ones that are in the ground and for conditions that they're not happy with, they turn into spores. When they come back, when their favorable conditions are now available, water or whatever temperature, now they wake up again naturally. Are their efficacy going to be the same as um, a brand new batch? Or would they lose efficacy if they wake up, let's say, 2 months later, 3 months later, a year later?
I think I understand your question, and whether you're talking about, i.e., native endospore forming bacteria that have been in the ground. Mechanism of action is mechanism of action. So I don't know if because they've been in the ground and then they're activated later on, if they are even more efficacious than a product where you apply the activated endospore forming bacteria. The point is with biologicals and biostimulants and plant production is the timing, not only the method of application, but the timing. So there may be endospore forming bacteria in the soil. A farmer does not have time. Blueberry formation, 30 days, 90 days, okay, before we get to fruit set. Cotton boll set, 40 days. So having a biological product that can deliver the endospore forming bacteria, irregardless of the quote unquote native bacteria in the soil, unless you have a way to trigger that real quickly, you don't have time. Agriculture is all
Great
about
answer.
timing, and timing is money. So, yeah,
That's what I
I
wanted
don't
to
know.
bring
Yeah,
light to. I totally 100% agree. For instance, the product that you put in today, you might have a little benefit next year, but very little, and you don't
exactly.
have any time to risk. So my
Exactly.
personal recommendation is that you use this product according to the advice of the company, the manufacturer, StrongSoil.
Exactly.
And so I totally, in my experience, and I have quite a bit, 14 years of it, I totally agree. Strong soil and the microbes in there, you want to keep putting product at a certain rate, at a certain time, but constantly is more important than, oh, well, I'm going to wait 6 months before I do it again. You're going to lose a opportunity to get all the great benefits that you get. So I just wanted to try to lead you into that answer so that the other folks could also understand it.
Thank
Thank
you.
you
Very
very
good.
much, Lynn. You're
Thank
amazing. I
you.
love you already.
Thank you,
Thank you.
Guillermo.
We did not pay you to say that, but I'll send you a hat.
No, he didn't pay me. I've been listening in. Uh, I've never met these folks, and I got a little experience myself, well, quite a bit. But yeah, I like what I'm listening. The different microbes that you have in there, amazing. Are you familiar with the Rhodosudomonas palustris?
No, I'm not.
Okay. No, it's just a photosynthetic microbe, also nitrogen fixing, and just
Yeah.
a pretty interesting little fella. Anyway, thank you so much.
Thanks,
Thank you,
Guillermo.
Guillermo.
Lynn, thank
Thank
you so much
you.
for doing this. Thank you for everyone for joining. I'm sorry if we didn't get to every question, but out of respect for time, we're gonna stop this now. We're happy to follow up. We will be also posting this in terms of a recording following this, so folks can definitely follow up. As Lynn mentioned, Lynn's obviously in business for herself, so if you wanted to contact Lynn and use her services, you are very much welcome to. We've been a very happy customer for the last 5 years. So very grateful also to Lynn and your expertise. We couldn't do this without you. So thanks again to everyone, and hopefully you have a great day. Take care.